Welcome to the Proteomics course. Today we will talk about surface plasmon resonance. We will have a discussion on Biacore SPR technique and data analysis. The biological systems depend upon the molecular interactions of two or more biomolecules, so that they can form the stable complexes. For biomolecular interactions, the principle of thermodynamics, biomolecular structure and recognition play a very crucial role. The identification of interacting protein and protein partners of the known function with those having the uncharacterized role. By performing these type of experiment, it is possible to understand the biological process of uncharacterized proteins. The ability to screen large number of proteins simultaneously and rapidly for biochemical activity, interactions of protein and protein, protein lipid, protein nucleic acids and small molecules require various high throughput instrumentation. So, currently the most popular methods which are used for detection of protein-protein interactions include yeast-2 hybrid and protein microarrays. As we have discussed in the previous lectures, that protein microarrays, they are one of the very robust techniques for studying protein-protein interactions, but these microarrays require label based detection system. Often these are fluorescence based detection. However, the label based detection techniques have certain limitations, such as the tags such as fluorescent tags may interfere with the function, including binding to the interactors and adding them to the query is always not so straightforward. So, there is need for label free biosensors which can avoid these issues and allow for the real time measurements. So, in today's lecture, we will have a discussion on surface plasmon resonance, and I have invited a guest to discuss about the Biacore technology as well as how to perform some data analysis using commercial softwares. So, now let us discuss about surface plasmon resonance. This is one of the very efficient and emerging label free technique for studying biomolecular interactions. The surface plasmon resonance biosensors are optical sensors which can exploit the surface plasmon polaritrons, the surface electromagnetic waves that can propagate parallel to a metal or dielectric surface. SPR is used to probe interactions between an analyte in solution and a receptor that are attached to the SPR sensor surface. Binding of molecules in the solution to the surface immobilized receptors changes the refractive index of the medium near the surface. The change in the refractive index of medium can be monitored in real time to measure accurately the amount of bound analyte, its affinity for the receptor and the association and dissociation kinetics of the reaction. Over the past decade, the SPR biosensor technology has made significant advancement, and a large number of SPR sensor platforms, the biomolecular recognition elements and measurement formats have been developed. The major strength of SPR biosensors are their versatility and ease of use. The SPR allows the analytes of receptor ligand interaction for wide range of molecular weights, affinities, and demonstrates the compatibility for small molecules and other chemicals. The SPR biosensors have played a very important role in biological research into biomolecules and their interactions, and now they are increasingly being used for detection and identification of chemical and biological interactions. Now, let us move on to the Biacore SPR technology. So, there has been growing interest in commercialization of SPR biosensors, which has led to a number of systems available in market. The commercial instrument from Pharmacia and then Biacore became available in 1990. Biacore are optical biosensors which can be used to monitor macromolecular interactions in real time without need to label the biomolecules. The Biacore is a versatile platform to determine the kinetic rate constants for a variety of interactions. A number of commercial SPR biosensor instruments are available. Since then, however, the Biacore system still dominates this field. So, let us now discuss about the Biacore technology with Lalit Kishore, the business leader research products in GE Healthcare Life Sciences, who handles the Biacore system. Hello.
Hi.
It is a pleasure to introduce Mr. Lalit Kishore from GE Healthcare to discuss about Biacore surface plasmon resonance technology. Lalit Kishore is business leader research product in GE Life Sciences, and today we will discuss about the surface plasmon resonance technology and its various applications in proteomics. So, Lalit, welcome to the discussion on SPR technology.
Thank you. Thank you.
So, how long have you been associated with Biacore SPR technology with GE Healthcare?
I've been with GE Healthcare for the last 12 years, and I've been associated with Biacore for the last 4.5 years. Been associated with Proteomics for a slightly longer time, for about 7 years now.
Oh, great. Okay. So, can you just brief us about your experience of using the Biacore technology from last several years?
My educational background is that I'm not a biologist, okay. Though Biacore is largely a biological tool. My background is in chemical engineering, so I'm a chemical engineer with an MS in management. However, I find very varied uses for SPR technology these days. I find its use in biopharmaceuticals, I find its use in basic research, in nanotechnology, in pharmaceutical industry, in QC labs. So basically, I think it's my chemistry and chemical engineering background that helps me work with Biacore, and quite a lot of applications that I see across the country on Biacore technology.
All right. So, you are utilizing your varied background to apply on the different biological problems.
Yes, that is what I think. SPR technology also does that, actually uses a very simple technology and applies it to different things in biology, and actually gives out results that are there for everyone to see.
So, Lalit, can you please tell us how you got interested in working on the surface plasmon resonance technology?
Yes. GE Healthcare has been associated with Biacore as a company for a very long time. In the year 2006, we actually acquired Biacore. And up until that point, Biacore was there only in some places in India. So I got into Biacore in 2006 sheerly out of interest in chemical interactions and interaction analysis. And ever since, I've been just working on Biacore and label-free interaction analysis technologies.
So, can you mention currently what are the major applications of SPR in the area of proteomics?
Actually, very vast very wide-ranging applications of SPR, starting from a simple binding analysis or kinetics analysis, analysis of affinity of interactions, whether it is protein-protein interactions, protein-DNA interactions, protein-RNA interactions, protein small molecule interactions in drug discovery, quality control, varied applications of SPR. Actually, we support a very large variety of customers from different backgrounds who want to do SPR in their labs.
Okay. So definitely you can see probably that SPR application will be very broad.
Yes.
In almost all the proteomics laboratory.
Absolutely.
Depending upon their experiments and the questions
Yes.
they want to ask.
Yes.
So Biacore is one of the pioneer in the field of studying the label-free interactions and studying about biomolecular interactions. Can you tell some of the latest advancement? What are the major applications by using the Biacore technology?
Let me start with a video that shows basically what SPR is.
Sure.
And then probably followed by another video of how biological analysis happens on Biacore. And then just a few videos, short ones, on how Biacore works. Okay. And then I'll quickly come to the applications of Biacore.
Okay, so we can
all right, let me show you the first video. So what I show here is the basic SPR phenomenon, right? This is the SPR chip, which you see here. On the top of the SPR chip is a gold layer, and on the top of it is a flow cell. At the bottom you see a hemispherical prism. So SPR phenomenon is pretty simple, that when you actually have the prism and you shine a laser light through the prism, as you will see shortly, when you shine the laser light through the prism, the light reflects at a total angle, angle of total internal reflection. The light reflects at the angle of total internal reflection, and evanescent energy waves are created on the top of the chip. And these evanescent energy waves are also called surface plasmons. And these surface plasmons are the ones which are used to actually study biological interaction. When I go to video 2 now, you will see how biological interactions are studied. Let us assume that you have an interaction, A plus B gives AB. What you do in SPR is you take one of the interactants, say B, and put it on the chip and pass A over it. Let me show you how it happens. You take one of the interactants, which is B in this case, and you actually immobilize it on the chip. You can see the molecules getting immobilized right now. And then when the molecules get immobilized, there is an increase in mass which changes the refractive index, and that is measured in real time. Now you pass the second interactant A over it. If binding happens and AB gets formed, you see a further increase in mass, which is again measured in real time. You stop the flow of A and start flowing a buffer, it comes off in a dissociation, and that dissociation is also seen in real time. So essentially what you are doing with Biacore is actually just measuring the amount of mass on surface of the chip. The mass on the surface increases or decreases, and that increase or decrease is measured in real time. This phenomenon is what we apply now to study biological interactions. Actually, Biacore is BIA for biological interaction analysis. So let me show you a few videos, examples of how Biacore can be used to study in some experiments. This is an example where A plus B gives AB. If you see the curve, and if the curve is existing, it means AB is formed. If you pass A over B and there is no response, which means if you see a flat line, then AB is not formed. So it's a very simple example where you can actually decide whether the interaction is happening or not happening. I will show you another example now of kinetics analysis, where you will see two examples. The first example will be that of an extremely rapid association. So you see the slope of the curve goes up very fast and comes down extremely fast. So this is a rapid association and dissociation. Whereas if you see the slope of this curve that is about to come up, it is very slow association and very slow dissociation. So just by looking at these curves, you are able to actually tell if the interaction is fast or if it is slow. So these are some of the examples of Biacore technology at work.
Right. So I think unlike microarrays where people can detect the interaction, but they cannot tell the nature of the interaction. Here the edge is that by looking at these type of kinetics and the curves, one can also tell about the type of association, dissociation, and the overall kinetic analysis.
Yes. So, and here I have on my PPT a very simple analysis of different things that can be done with Biacore. So when someone asks me what can be done with Biacore, these are the 6 things that can be done with Biacore. So very shortly put, these are the 6 applications of Biacore. For whenever A plus B forms AB, when you're studying an interaction A plus B gives AB, the questions you ask is the first question you ask is, does the interaction happen or not? Which means, is the molecule AB formed or not? The second question you ask is, how fast is the interaction or how slow is the dissociation? The third question that you ask is, how strong is the interaction? So what is the affinity of the interaction? The fourth question you ask is, how much of the analyte is there? Right. Which means, what is the concentration of this analyte? Sometimes in drug industry you ask, is this interaction a safe interaction or not? Yep. And sometimes if you have a heterogeneous analyte and you see that the binding is happening, you want to ask, what is it that is binding? Because there are too many components in this analyte. So what is it that is specifically binding? Right. And these are the 5 different things that you can do with Biacore.
I think as you rightly mentioned, identifying the very specific interactor is the most challenging aspect of it.
Yes.
Because that's where many times people fail and they discover the false interactors.
Yes.
So I think that's where the SPR has edge over conventional techniques like immunoprecipitation or yeast-2 hybrids. Yes. And some of the other large screening methods where there's a good chance of identifying the false positives.
Sure. There's a lot of promiscuous binders in screening experiments, right, which will be avoided if you have the specificity that Biacore gives you.
Sure.
So we start with each of these applications in detail. The first application which we will talk about is the specificity application. Now, some interesting questions that are asked when you're doing specificity applications. Is the drug binding to the receptor or not? Is the mAb identifying a strain or not? Or is there any non-specific binding in the interaction that I'm studying? And these questions are very easily answered by Biacore. Shown here in my PPT is actually an example where we're looking for binding. And here, someone came to us with about 40 different compounds, and they want to see if any of these compounds binds to a receptor. And here we have 40 of these experiments done. Most of these are not binding or binding at a very base level. But if you look at this presentation, I've highlighted one spot here with one molecule that's shown circled in red. This particular molecule is actually binding to the receptor. So at the end of a very short experiment of looking at the receptor versus candidate binding, you're able to determine which of these candidates is actually binding to the given receptor. A very simple example of specificity. Going on to the next application.
So, sorry, I'll interrupt you here. So basically, they'll just demonstrate that even if we are very much unaware about the components which could be interacting, this could be a good screening tool, right? Because
absolutely.
If there is a real strong interaction and a specific interaction, then probably we can see some outliers like these.
Absolutely. If you want to do that first level screening to just quickly find binders, right, in some kind of a screening experiment like you mentioned, then this would be a very good starting point for you to quickly find those binders and take them to the next level.
Right. Many times when we have a thrust of identifying some like large drug library, right,
yes,
or small compounds, and then at that time maybe to begin with, this
absolutely
could be a good screening tool.
Yeah. I would also like to point out that this is a very small experiment. It barely takes about 1 minute to do this experiment. So basically per minute you can do one screen, and that way you will have a lot of compounds screened in a very rapid manner. So that is the first step in the specificity experiments. Now let us assume that you found this candidate and you think that it is a specific binder, and now you want to look at the kinetics of the interaction. And everyone understands that kinetics is an extremely important part in drug discovery. In proteomics, when you look at interactions, one of the important things that you need to look at is what is the on rate and off rate of the interaction. Absolutely. And so in that sense, how fast does this interaction happen, and which candidate is kinetically preferred? Because if you have two candidates, both of them trying to be drugs, you should choose the candidate that is kinetically preferred. And in most cases, in recent cases actually, kinetics is being used to show similarity of drugs. For example, there's a lot of biosimilars coming out of India, and biosimilar manufacturers actually want to show that their molecule is similar or equal to the innovator's drug. Then one good way of showing that they're similar is by showing the similarity in kinetics. So that is where again kinetics experiments will be helpful to you. Experimenters, scientists can actually calculate the K on, K off, or the Ka, Kd, and the K capital D, which is the affinity of the interaction. They can calculate this and in a very fast way actually understand the interaction a little better than they did before. Because in the first instance, they only knew whether the interaction was happening or not. Whereas right now, they also know that what the kinetic parameters of this interaction are. And once they know the kinetics of the interaction, they come to affinity.
So identifying the on rate, off rate, and dissociation constant, I think this provides a very strong tool and information
for characterization.
And that is, I think, the big thrust of all the pharmaceuticals for doing biosimilars.
Absolutely. Anyone who is pursuing biopharmaceuticals or recombinant proteins, biosimilars, or even novel drug discovery people in the small molecule arena want to actually characterize the interaction in terms of the on rate, the off rate, and the affinity of the interaction. And that's something that is very well accomplished by the SPR technology. So when it comes to affinity, as you can see here, how strong is the bond? Is the binding strong enough to be physiologically important? And this is one very important thing, because more and more drugs are coming out these days which are one dose a day drugs, or which are fast acting drugs, and these kind of discoveries depend a lot on kinetics and affinity of the interactions. And that's where Biacore can actually come in a very big way and help people genetically or protein engineer their drugs so that they actually perform better than existing drugs.
For a good comparison with the existing.
Absolutely. To make drugs better, or to discover novel drugs that actually act better. Both of the cases, I think SPR technology can be useful. Once you know the affinity and concentration, and I always think that concentration is very less studied, but it's very important, is because if you look at concentration analysis across the world for proteins, there is no way that someone can measure active protein concentration without having calibrants in their hands. Whenever you give a student a protein and say, please measure the concentration, the first thing that he will ask you is for calibrants. He will ask you for standards. And the problem with these standards is that sometimes they're not available, sometimes they're extremely expensive, and being proteins, like sometimes they degrade.
Not very stable,
They're not stable. So there is a great need for having a concentration analysis technique that does not need calibrants. And that's where Biacore comes in again. Biacore does something that's called CFCA, which is calibrant-free concentration analysis. And so within 5 minutes, if you have a specific binder for a protein, you can actually calculate the concentration of that protein without the need for calibrants. And since you already seen in screen 1 where we talked about specificity, we are talking about specific binding. So what is measured is not just total protein concentration. What is measured is specific active protein concentration.
I think that's a very strong application of it, because many times you'd like to know how accurately you can determine the protein concentration.
Absolutely, especially in quality control and in filling. In biopharmaceuticals again, where people need to exactly estimate how much they are actually filling in the final vial that actually goes to the patient, they need more accurate methods of measuring active protein concentration. And that is where I think Biacore will play a very big role in letting people estimate that active protein concentration. Now, once you've seen concentration, the next thing is immunogenicity. Worldwide, with the increased biopharmaceuticals, drugs everywhere need to be tested for immunogenicity. Immunogenicity is about direct measurement of anti-drug antibodies in the which should be measured in serum. It's also about bringing a regulatory framework into a system. Right now, we talked about only technology and science, but suddenly when it comes to drugs, regulators come in. So can we actually accurately, confidently measure anti-drug antibodies in animal sera or in human sera at clinical trial levels. And Biacore can actually be used for accurate measurement of anti-drug antibodies and for immunogenicity testing of biopharmaceuticals. So that's another major application of Biacore as it comes in. The last application of Biacore, which if you remember when I talked about in the first slide about specificity, now if you have a heterogeneous mixture that is flowing over a ligand and something within that mixture is actually bound, and you see a curve and you know that something has bound, but you don't know what it is that is bound. So you can use a technique called SPRMS, where you can take the bound analyte, separate it into a vial, and then actually take it to a mass spec and identify the protein. So in addition to doing all that it did before, now you can actually also find out what is it that is bound.
So definitely it's very important, because many times you will not know like what the unknown target is, which is interacting or binding, right?
Yes. So if you don't know, if you have this unknown target, for example, if you have a receptor and if you have cell lysate,
right,
or if you have some kind of a homogeneous tissue lysate,
right,
which you are flowing over the ligand, and now you can actually find out what that is. So it is used in applications like ligand fishing, for example, where you're fishing for a ligand.
Mm-hmm.
So those are the kind of examples that.
But in MS, you will definitely require protein above certain threshold, right? So how you overcome that issue, because the binding will not
be
give
minimal.
You enough protein.
So one thing is that this is a very small interaction happening. So the amount of protein that you collect may not be sufficient. The only way you can overcome it is by actually doing it multiple times,
okay,
collecting enough so that you can actually get some kind of an MS response. And that is what most of our users do, is that they run the same binding assay about 10-20 times
mm-hmm.
and collect the bound analyte and then take it to an MS and then get their result.
So in Biacore, you have a way to collect the flow in the flow cell.
Yes.
And then actually you can accumulate that in multiple
runs and then take it.
Then concentrate that, and then you do further
yes.
characterization.
Yes. So just to summarize, again, I will just go back to my the first slide that I showed here, which is here. These are the 6 things that a Biacore can be used for: specificity, kinetics, affinity, concentration, immunogenicity, and mass spec analysis. So those are the 6 broad applications of Biacore SPR technology.
I think those are very important, because many times a good decision about these products beforehand, about characterizing these proteins, can save you a lot of money, a lot of effort down the road, because
absolutely,
if you can do those experiments in the beginning.
Most often when we actually talk to pharma industry, we tell them that Biacore or SPR technology is not for success but more for failure.
Right.
Only thing is that we say that it is for early failure and it is for cheap failure.
Right.
So do not spend too much money on something that does not work.
Right.
Might as well fail early.
Right.
So that is the basic reason why SPR should be used.
I think that's very important. So can you briefly tell us about what are the major instrumentation available from the Biacore technology currently for doing the surface plasmon resonance based experiments?
There are basically 4 different Biacore instruments available. There's a very small Biacore which is called the X100. You can see it on my pitch. It has 2 flow cells and it can do some beginning analysis. And then you have the Biacore 3000, which is an academic favorite. A lot of academicians like it. A lot of customers in India who are academics have the Biacore 3000. Then we have the latest, which is the Biacore T200. Very special again because it has all the things that the Biacore 3000 has, but it is regulatory approved. So if you are a company that works with FDA, that works with EMEA, that works with DGCA or some of these regulatory authorities, then I think that you should be using the Biacore T200. The Biacore 4000 has 20 different immobilization sites on it, and Biacore 4000 can be used with multiple 384 well plates with robotics. So if you're a company that has extremely high throughput screening, if you're a company that does a lot of immunogenicity experiments, or if you're doing batch testing or release testing using Biacore, then you should be using the Biacore 4000. I must say that most of the customers in India use either the Biacore 3000 or use the Biacore T200.
So how easy it is to do these SPR experiment, and especially the kinetic analysis by using some software available from Biacore?
The the most important thing in a Biacore experiment is actually the experiment design. It's very easy to do the analysis, and let me just show you an example of a typical result.
Okay.
And let me show you how the analysis works out.
Sure.
So if you can see my computer, so I'm going to open a typical Biacore result, and here is a Biacore result. This is a Biacore result where 5 samples of different concentrations were run over a fixed ligand which was on the chip, and now I'm trying to do kinetic analysis. So if you look at these 5 results, I select them and I show the results, and these are the results. Now this is what a typical result looks like, and there's nothing to be worried about because it looks odd. The idea is that your results are all embedded somewhere here. These 2 big peaks that you see are regenerations. So the first thing that I do when I do the analysis is actually select the regenerations which I don't need and cut them out. So I cut them out here. So I say cut, and then the rest of the results.
So you want to adjust your response axis.
Just so now I have 5 different concentrations, 1, 2, 3, 4, 5 in different colors, and you have, this is the association phase and this is the dissociation phase. Now I'm going to just baseline this result. So I select the baseline here, and then I just go to the adjustment of y-axis and I say 0 at the average of selection, and then I say add as new. And now all my 5 results are shown here. Now I'm going to do a quick kinetic analysis, and it's really extremely simple to do a kinetic analysis because all that I do is say calculate, I say kinetics, simultaneous K on and K off, and I have already done the cutting and the Y transformation, so I say next. And then if I want to, I can go and adjust the start and the end time. So I can actually move this to adjust the start time and the end time of association and dissociation, which I sometimes do, but I think this is pretty well picked up by the software already. So I don't need to do much. I say next, and I enter the different concentrations of each of the samples which were run. They're entered here. Now this is very important.
These concentrations are known concentrations
which you ran
already
on,
started.
which you actually made the concentrations of the analyte and then ran them over the ligand.
Sure.
So you would do that. Now one of the things that is important here is you choose the model. So preferably it is always better whenever you do characterization, the more you know about your system, the better characterization results you get.
Right.
So in this case, suppose you do know that it's a 1-to-1 binding, then you would choose that binding. But if you want to, you can actually change the binding model. It could be a bivalent analyte, it could be a bivalent ligand,
Right.
it could be a heterogeneous ligand. So depending on the model you choose, so you choose the model that you want to here, and then you say fit. And what happens is curves get fit and the results are thrown up. So your K on and K off are displayed here. So it's as simple as that. So all you need to do is take a ligand, immobilize it on a chip,
Right.
run about 5 different concentrations of your analyte over the chip, and this simple and each of these results, if you look at the x-axis carefully, it's 0 to 600. That means each one of these runs, the entire run
Right.
was 10 minutes,
Yeah.
and you ran 5 samples, so it was 5 into 10 minutes, so 50 minutes, with the time taken in between the runs about another 10 to 20 minutes, about 1 hour 10 minutes, 1 hour 20 minutes, you have the results, and you have already characterized your results because you have the K on and K off calculated. And you can also quickly check how good your results are by quickly checking the residuals, and you can see the residuals here. The chi-square values are really between -5 to +5%. So extremely good
Very tight.
fitting reactions and a very fast analysis that gives you K on and K off.
Yeah, I think it's very extremely fast and very good. way
Very
of
easy to
analyzing
use, very easy to use
interactions.
software for your analysis.
So yeah, it was very useful and informative to see the analysis, like how easy it is to perform the kinetic analysis. Can you give some specific example of doing kinetic analysis by using Biacore system?
Let me show you one example, and this again goes back to why kinetics is important. Kinetics is extremely important in Biacore analysis. And let me show you an example. So here's a pitch, about 3 slides on kinetic and affinity analysis using Biacore. So if you look at this slide, now here on this slide, you have 3 interactions which are captured. You have interaction 1 which is captured in blue, interaction 2 which is captured in red, and interaction 3 which is captured in black. The important thing about all these three interactions is that all these three interactions have the same affinity. They have varying K on and K off, but they have the same affinity. And this is important in drug discovery because let us say, for example, you were looking for pain relief. If you want pain relief, you want a drug that acts fast and that stays on for a very long time. But if you want a sleeping pill, for example, you want a drug that actually acts slowly but stays on for only a reasonable amount of time and comes off fast enough. So kinetics is very important in choosing a drug candidate. Now this is an example where if someone chose just on the basis of affinity, all these three would have been the same. But since they would make their choice based on kinetics, they can actually decide based on the K on and the K off. Now here is a real life example in the next slide which I show you, which is a publication from Amgen and from David Mishka of University of Utah, where actually Amgen uses this data directly to do their clone selection. So they're actually doing MABs, selection of MABs, selection of clones for MABs. And if you look at this, so clone number 1 and clone number 2 have similar affinities, but if you look at their on rate and off rate, there's a 10-fold difference. So if you look at these two clones, for example, which clone should they go for? They should go for the clone which is having more kinetically relevant properties. If they use only affinity for making that choice, then there is no choice at all. They're both the same.
Right.
So this is a live example of where kinetics data is actually being used to capture information regarding K on and K off and then make a more educated, knowledgeable decision on which map to go forward with.
Right. I think it's very interesting to see this because you will feel that if you just rely totally on the KD values, right? If you do not go individual on the K on and K off.
Yes. So distribute that KD value into K on and K off and make a more knowledgeable decision. Yes.
So it was a very interesting example to appreciate the power of kinetic analysis, right? Can you just brief us about what are the major limitations or shortcomings of various surface plasmon resonance based technologies?
Absolutely. So like any technology, this technology should not be viewed as a silver bullet. It does have shortcomings. One of the significant shortcomings being that if there is any structural differentiation in the protein, this would not be able to capture it. It's a mass-based sensor, so any structural changes will not be captured. There is also a problem that there are situations where you are unable to immobilize the protein that you have onto the chip.
Right.
So you might have to use some capture techniques to do that. There is also this big question about, so you answer these questions about interaction, you answer questions about whether the interaction happens or not, how fast, how slow, how strong, how much.
Right.
You say whether it is safe or not with immunogenicity, and you know what is binding, but then sometimes you want to ask the question, why is it binding?
Right.
And why is it binding is answered by structural studies or thermodynamic studies. So that is where I think Biacore can give you a little bit of a direction, but I think you should do more of an NMR study, or you should do microcalorimetry, and that's what will probably give you more answers on why the interaction is happening.
So further in-depth studies will be required on those
Absolutely.
interactions.
So when you have an interaction happening and when it's happening very fast.
Right.
Now why is it happening fast? Is transition state thermodynamics. So you will have to ask questions in thermodynamics to get those answers. So sometimes further study is essential, and Biacore kind of stops with these applications.
Would you have any final advice, take home to the
Absolutely.
students?
So here's basically 3 rules that we have in Biacore. The first rule in Biacore experiments is Biacore technology is extremely easy to understand. Biacore technology is extremely easy to analyze. A lot of time must be spent on experiment design. The first thing that I would do if I were working with Biacore is spend a lot of time on very carefully considered experiment design. The second thing that I would do if I were conducting a Biacore experiment is that I would make sure that I have an extremely pure ligand that I will put on the chip, and that is extremely important. The last thing that I would consider, and this is true of all experiments, and I'm sure you would agree with me, is that it's garbage in, garbage out. So make sure your sample preps are correct, and remember that this is an analytical instrument. Unlike many other techniques in biotech, this is an analytical instrument. It is a mass sensor. At the end of the day, you could call it an extremely sophisticated weighing machine. So if you put something on it, it will give you the weight. It's as simple as that. Only thing is you have to do it right. So I would say that make sure that your sample prep is perfect, your experiment design is perfect, make sure your ligand is pure.
So I think you very rightly mentioned that a good experimental design, identifying the good ligands, working out the chemistry for immobilization,
Absolutely.
as well as doing the very good sample preparation. All of these are very essential component for doing any successful proteome experiment, and especially the surface plasmon type of experiments.
Yes.
So thank you very much, Lalit, for discussing about the Biacore technology with us today.
Thank you.
And I hope it is informative and useful for my students.
Thank you. Thank you.
So after having a discussion with Lalit Kishore on Biacore SPR technology, I hope now you are very clear about the instrumentation, the various properties which can be studied by using the Biacore system, and how quickly one can perform the data analysis. So conclusion from this whole discussion is that since the introduction of the Biacore SPR instrument, the SPR spectroscopy has become widely used from chemistry and biochemistry to characterize biological surfaces and monitor the biomolecular binding events. Overall, the success of SPR technique is due to the following factors. First, the kinetic measurement in real time, that is a major strength of data obtained from the SPR instruments. Second, monitoring the adsorption of unlabeled analyte molecules to the surface. And third, its ability to monitor weakly bound interactions due to high surface sensitivity of SPR biosensors. So, in summary, in today's lecture, you have learnt about significance of studying the biomolecular interactions, how surface plasmon resonance technique can be used for studying the biomolecular interaction, a brief introduction of Biacore technology and data analysis, and then certain issues and challenges which are associated with SPR technology. So, we will continue our discussion on label free techniques in the subsequent lectures. Thank you.